So when you're thinking about the kind of germline genetics, kind of the first basic question to always ask in Gen Epi is inherited genetics actually generally important? So of course, the answer to that question is pretty much always yes, but you have to ask it anyway. And we know that ovarian cancer tends to run in families. There's lots of data to show that, and I'll show you a bit of that. And twin studies, so like most other cancers, twin studies have told us that most of the kind of heritable component disease risk is due to shared genetics rather than shared environment within families, although shared environment within families is important. And so sort of early on in my PhD, I did a meta-analysis looking at the familial risk of ovarian cancer. And like most cancers, if you've got a first-degree relative with that cancer, you're at increased risk. And for ovarian cancer, the familial relative risk is approximately 3, a little bit bigger if you're younger, or if your relatives are affected at younger age, a little bit smaller if you're older, or your relatives are affected at an older age. So we know that inherited genetics is important, but then you kind of ask the question, what's the genetic architecture of disease? And when I talk about the genetic architecture, what I really mean is what's the range of risk allele frequencies, the allele frequencies in the population, and what are the risks they confer? And the underlying architecture of different phenotypes is fairly different, but broadly speaking, I think about kind of rare variants, uncommon variants, and common variants. But of course, there's no absolute definition of what that means. But rare variants maybe less than allele frequency of say 0.5%. So rare variants could confer high risks of disease. They could also confer moderate risks of disease and have weak effects. So we published our first genome-wide association study in ovarian cancer in 2009. It's interesting that in those days, you find one SNP associated with ovarian cancer, you've got the Nature Genetics paper. One ovarian cancer SNP might be published anywhere these days, apart from one of those predictive journals that we pay them to publish. But anyway, in those days, we got a Nature Genetics paper. And so that was our first GWAS. Basically, it was based on 2,000 cases of 2,000 controls. We marked out one hit. So kind of the next 10 years, I've been in the industry of GWAS in general, of ever increasing sample sizes, and ovarian cancer is no different. Part of the COPS project, which was a multi-cancer European Union funded project. We ended up having 18,000 ovarian cancer cases, 26,000 controls. Identified 11 variants associated with ovarian cancer. Then the Game On or OncoRay project, which was even more cancer consortia put together, funded mainly by the NIH, which was sample new funding. We increased our sample size to 26,000 cases and 40,000 controls, and we ended up having 34 SNPs for ovarian cancer, and we then started to do meta-analyses of multi-cancers, variants that were shared between different cancers. So by 2017, we got to 40 variants associated with ovarian cancer. That's pretty much the history of GWAS and ovarian cancer, more or less where we are today. We are in the process at the moment of what we've been doing is taking all these original data and re-imputing them to better and better imputation panels and squeezing more and more out of it, but we've really got much beyond that. And as you can see, we get Nature Genetics papers, and it starts going down with Nature Communications papers, and then it becomes something else. So we publish lots and lots of these in relatively good journals. So just to go on to sort of the timeline for all of this. So this graph is time on the x-axis, and the bar plot is the number of SNP-associated disease, which is the blue line. And then the red line is the fraction on the component of familial risk explained by these variables. So the first variant we discovered, as I said, in 2009, that's one variant on the x-axis, and that explains about 1% the genetic component of disease risk. That's why it was the first one we found. Relatively big effect, relatively common. And then those data from the first GWAS ended up with a couple more publications, with a handful of more SNPs over the next few years. And then we published the first Fox papers in 2013, so that was the first big collaborative multi-cancer effort, EU funded, and we found a whole handful more variants over the next couple of years. And then the variants explained going up to about 4%. And then the OnCarray, before array, we did a meta-analysis of Ovarian Cancer Association sorting data and sorted investigators and modifiers of BRCA1 risk, which are both ovarian cancers, found a little bit more. And then there was the OnCarray, which we published in 2017, taking us up to over 30 variants and explaining just over 5% of the minimum compassionate disease risk. That's kind of been progress GWAS has been over years, and just to put that 5% onto our little pie chart. So about a third explained by BRCA1 and BRCA2. Now all this GWAS ever explaining 5%, so a little slice of the pie. So the hungry people have BRCA1 slices, and the hungry various slices of the pie. But of course, we know that there's more common variants than those we've found, so we're limited by sample size and effect size of these variants. And there's something called the chip heritability, where you can estimate the proportion of the heritability or the component of disease risk that is explained by all of the common variants that are captured by the genotyping arrays. So we know there's actually thousands of common variants. You just can't detect them at very strange levels of significance. And so you can do that calculation. It's a relatively straightforward calculation. And so for ovarian cancer, we can say that although genome-wide significance, we've only got 5% variants explained, the common variants actually probably explain about 40% of the inheritance component of disease risk. So if you then put that on your pie chart instead, now we've got a third of rare variants and 40% common variants. So we can have this little chunk of pie here that is due to other rare variants, not BRCA1, not BRCA2, uncommon variants that aren't captured by the genotype arrays. That was the middle row of my genetic architecture and pie plus possibly some rare variants with moderate effects. So at this point, I just wanted to say something about polygenic risk and ovarian cancer, because once we've discovered all these common variants, you start thinking about how do they work together. If they're common, people will carry more than one risk allele at these different variants. And I put this slide up because it's ovarian cancer, but I first started thinking about this a long time ago, published a theoretical paper 2002, suggesting that actually if we found lots of these common variants, it might have implications for disease prevention. This was focused on breast cancer. And in this paper, we sort of suggested that actually a range of risks, common variants might explainably be enough to be able to do targeted prevention, like targeted mammography of breast cancer. And the reason for this is that the polygenic model predicts distribution of risk in this population that was
log normal. In other words, the relative risk was log relative risk would be normally distributed population. That would mean that individuals without disease have a risk distribution that was then shifted to the right curve for individuals with disease. And this model has turned out to be more or less accurate for lots and lots of phenotypes. Just to try and explain how this polygenic risk arises and why it's log normal. So imagine we've got a single SNP and the allele frequencies are 50-50. So the risk allele is 50% and the non-risk allele is 50%. And so the number of risk alleles an individual can carry is 0, 1, or 2 because we'll carry 2 copies of every allele. And the frequency of those risk alleles is what you expect from a high Weinberg would be 25%, 50%, and 25%. That's very simple. How does that then relate to risk? We've got a number of alleles on the x-axis here. How does that relate to risk? So here I've got an example genotype where the 2 alleles are A and C, and we're going to say the C allele is the risk allele. So the 3 genotypes are AA, AC, CC. So you either carry 0, 1, or 2 risk alleles. And in genetically, what we used to do was calculate the relative risk, well, for the case control study, the odds ratio of the heterozygotes compared with the non-risk allele homozygotes. Let's call that little r. Then we calculate the relative risk, which would be just a single big R of the risk homozygotes compared to the non-risk homozygotes. And under a codominant model, what we expect is that RR will be R times R. So they're just multiplicative risks. And it turns out, I discovered this morning about this, that the codominant model really works pretty well. When we first started doing this, we didn't know that it would. But actually, the codominant model where we estimate little r from a per allele model works really quite well. We call that the per allele most risk. So most genome-wide associations use the simple model, but it has consequences. So it's multiplicative within a variant, but it also turns out to be multiplicative between variants. And so if you have n SNPs associated with risk, the number of risk alleles you can carry is anything from 0 to 2n. So in 40 risk SNPs, if we have ovarian cancer, the woman can carry anything from 0 to 80 risk alleles. That's just the number of alleles, it's not the risk itself. So if we now look at our histogram of the number of risk alleles carried, this is going to be a video going through increasing number of SNPs. That's what distribution of the number of risk alleles women in the population will carry. Yeah. Now, the number of SNPs increases, then the distribution begins to look like this. So once you get to 40 SNPs, then the mean number of risk alleles carried is 40. Some people might carry 80, but they're vanishingly rare. Some people might carry 0, and they're vanishingly rare, but this is a binomial distribution. A binomial distribution approximates to a normal distribution as n gets bigger and bigger. So in terms of the number of risk alleles, the number of risk alleles carried is normally distributed. And of course, that then leads to the fact that if all the risk alleles have the same relative risk, then the number of risk alleles, the log relative risk would add up, or the risk would multiply. So the relative risk would be little r, the power I, the number you carry. So the log relative risk is expected to be normally distributed. That's why you get this normal distribution, which is really useful. It has nice mathematical functions that we kind of understand, that we can predict. That's a simplified model where it assumes that all risk alleles have the same risk allele frequency and the same risk. Of course, that's not true, but it doesn't matter. Because we can calculate a polygenic risk, where if we've got multiple risk alleles, so each of these components of this formula represents different single nucleotide polymorphism. And for each one, we have a number, which is the number of risk alleles you carry that polymorphism, or 1 or 2, and then log relative risk, or log ratio, in your case control study, is the weight that SNP contributes to your polygenic risk score, and you just add those up across all your variants for ovarian cancer from up to 40 variants, but you can do this across thousands, tens of thousands, and there are polygenic risk scores out there with millions of variances, each one with very, very small betas, generating a polygenic risk score. And then you can calculate a relative risk, which is the polygenic risk score exponentiated because it's from polygenic risk to the log relative risk, so the relative risk is e to the power PRS. And in simple terms, the relative risk is always compared to something. That's the relative risk compared to individuals with no risk alleles at all, with W1 0 all the way out to W5 or 0, polygenic risk score 0. But I won't go into it, but you can rescale it. And so this is the polygenic risk distribution for ovarian cancer based on our 40 risk steps. And so on the x-axis is the log relative risk that I've exponentiated, so it's now relative risk on a log scale. And I've rescaled it, so it's not compared to people with no risk alleles, but it's compared to the average risk in a population. So the average relative risk on this is 1. You'll see is the average log relative risk is less than 1. That's median is slightly less than 1. Median relative risk is 0.95. And then the risks at the 5th and the 90th percentile of this distribution are about 0.6 and about 1.6. What does that mean for kind of absolute risks in ovarian cancer? So based on UK incidence data, that's a lifetime risk, median lifetime risk of about 1.7%. The 5th percentile is 1%, the 90th percentile nearly 3%. Whether they are clinically relevant or not, I'll come on to, probably not, I'm sure I'll say. And just to put that curve on a linear scale of relative risk, now you can see why the average is different from the median, because it's obviously a skewed distribution. There's this long tail of people at fairly high risk. But sort of corollary of the fact that it's a normal log relative risk is that most people are less than average risk, which was a bit hard to wrap your head around, but that's true if it's a skewed distribution. So one of the things that's turned out, the multiplicative model or the log-additive model worked really well for within and between SNP interactions. It also turns out to work really well for interactions between genetic effects and other lifestyle risk factors. So for ovarian cancer, there's a whole load of risk factors that we know about associated with ovarian cancer risk. And it turns out they all interact multiplicatively with the genetic risk factors. And what that means is you can just add them all together and get a risk distribution with a bigger variance or a bigger standard deviation. Very easy to do. And so you can now, what I'll do is just show you the table of the risk distributions. These are log normals, the variances. So the polygenic risk distribution has a variance of just under 0.1. And the risk factors have a variance of about 0.2. You just add those variances together because they're independent. So the variance of the combined risk distribution is about 0.3. And I put the relative risks of the 50th and 90th percentiles in the table. So you can see the widening of the risk distribution. The other point being that as your risk distribution gets wider, your median moves to the left. So your median goes from 0.5, 9 to 0.86. And then the 5th percentile risk gets lower, 90th percentile gets higher. But even with a combined model, 5% of women at high risk have a relative risk of just over 2 and above. So it's still not a huge relative risk based on everything we know. How do those cumulative risks based on the combined model, how does that transfer to sort of cumulative risk? This is similar to the penetrance of BRCA1 and BRCA2, but instead it's across the risk distribution. So what the dotted pink lines at the top and the bottom are at the 1st and the 99th centile. So women at the 1st centile of this combined risk was very low lifetime risk of less than 0.5%. Women at the 99th centile have a moderately high risk of ovarian cancer of about 4%. But that's a lot smaller than the nearly 20% risk of a BRCA2 carrier, for example, just to compare the two. And then in between, I've got the 20th, 50th, and 80th centiles as well as the average. So that's what polygenic risk combined with other risk factors can do for ovarian cancer. So I've talked about rare variants, BRCA1 and 2, talked about the common variants, where we've got to the common variants. So I now want to talk about the middle of my genetic architecture slide and say something about uncommon variants. So by definition, most of these are not tagged by genotyping arrays, although bigger and bigger imputation panels mean that we're starting to be able to look at uncommon variants from genotyping array data, but broadly speaking, not. And therefore, the only way to capture common variants is by sequencing. So sequencing remains costly. Genotyping is pretty cheap. You can do a genome-wide array for $20 or whatever it is now for a Illumina array. Sequencing is still in the tens, hundreds of dollars for one sample, so it's costly. And so the early association studies with sequencing are based on targeted sequencing of candidate genes. We could afford to go with exome or genome-wide. We could do targeted sequencing of genes that we thought might be interesting. And so we started doing targeted sequencing studies now about 10 years ago, because developments in sequencing technology meant we could actually do multiple genes in a single experiment, and the costs had come down sufficiently so you could do thousands of samples, not tens of thousands, but thousands. And we were using a platform called Fluidime that used multiplex PCR and barcode labeling of fragments to enable us to do large numbers of fragments in single experiments, and then sequencing technology. We have to do all of that sequencing and then to untangle the different fragments, the different individuals that were called into single samples. So we got the cost down to about $20 a sample, and we're able to do thousands. So we did a whole series of targeted gene experiments. We were focusing on DNA repair genes because BRCA1 and 2 are DNA repair genes, and we thought this is where most of the interest would be. So each of these is showing the different panels that we did with the genes listed. Obviously, I'm not going to go through every gene, but we started off by doing what we think were the most interesting ones, and then we expanded some of the ones that were interesting, like RAD51B, C, and D, into additional case control series. And so we ended up with around 6,000 cases and 6,000 controls that were being sequenced for most of these genes. And we were able to do the looking for rare variants in these genes, the coding sequence of these genes, and ovarian cancer risk. And then that long list of genes that we looked at, the ones that emerged So we already knew about mismatch repair genes in ovarian cancer because they had come out of linkage studies in Lynch syndrome families. It was clear that ovarian cancer was part of the phenotype of Lynch syndrome, so that wasn't a surprise. But we showed fairly strong evidence that loss of function mutation or variants in RAD51C and D were associated with ovarian cancer, relative risk of about 10. I'll show you in a minute, very big confidence intervals. BRCA1, BRCA1, not BRCA2. BRCA1, sorry. Also strongly associated, and then weaker evidence for pancreas. We now think that pancreas is probably more associated. We've done bigger studies and CALB2. So that's our list of genes now that we've got for ovarian cancer: BRCA1, BRCA2, common variants, RAD51C, RAD51D, mismatch repair genes, BRCA1, CALB2. And those genes are now incorporated in clinical genetics, the panel testing for ovarian cancer, and widely used. So what does that mean for our little pie? Well, they also explain about 5% of the variants. So now we've got to the point where we've done everything we can, and we've got to about 3/4 of the ovarian cancer variants explained, but there's still quite a lot unexplained. So what's the clinical relevance of all of this? Well, the first thing to say is, the options for clinical management are quite limited. We haven't got many tools in our armory for preventing ovarian cancer. And secondary prevention or screening, early detection, has really been shown not to be effective using things like CA125 and pelvic ultrasound. What we do know is extremely effective is prophylactic surgery. So if a woman has her fallopian tubes and her ovaries removed, her risk of ovarian cancer is reduced enormously. 95% is not, there's still some risk of developing peritoneal disease, but it's reduced hugely. But prophylactic BSO before the menopause is associated with surgically induced menopause, which can have quite unpleasant side effects, symptoms of surgical menopause, as well as obviously affecting individuals' fertility if women are still thinking about families and stuff. So BSO is not a fantastic intervention that can sort of explore everybody who's at reasonably high risk gets on with it. So what is not really well established is what's the level of risk somebody should be at before being offered. In the UK, it's offered to women who have a 10% lifetime risk. Lots of people say it should be lower than that. And there's a study showing it's very cost effective, even down to 5% or even 3% lifetime risk. So, but these thresholds aren't well established yet. So a BRCA2 carrier is kind of well above that level of risk on average, because it's kind of about 20%. Some of the genes like that have BRCA1, CALB2, RAD51, it becomes a bit questionable. So just to say something about RAD51 and 2 particularly, so it's well established for BRCA1 and 2, but for RAD51 and 2, RAD51C and D, the risk estimates are quite imprecise. So when we think about the risks, what that means for clinical counselling, we're thinking about our 10% threshold. So this, again, is a kind of skin to risk curve, and the solid blue line is the point estimate, the risk of RAD51C or RAD51D, which are both about relative risks of academic. If you put the confidence limits on those point estimates, but those risks, you could say to a woman with the RAD51 mutation, well, we think your risk is somewhere between 5% and 35%. That's kind of not a very helpful range, I think, from a clinical perspective. Well, what am I supposed to do with that? High risk or not, we don't really know. So what we do know is that we need even bigger studies. We've sequenced 6,000 cases of controls. What we need to do is sequence 15,000 cases and just get those risk estimates more precise. But that work is ongoing. We do know that we've got better estimates now, a bit more precise than this. But the other thing that sort of is ongoing and sort of work in progress is kind of comprehensive risk models. So, we've shown that we've got rare variants, common variants, we've got the uncommon variants, we've got lifestyle and environmental risk factors. We can put all of those into one model. So you can't do that kind of clinically because you can't put all these things in your head and come up with a risk calculation that's even, you need a computer model, risk model to do it. My colleague Anthony Santonio has been working on this since the early 2000s, using all of the data we generated. We originally created a BRCA model, which was focused on BRCA1 and 2, and this has gradually been expanded into a comprehensive risk model for breast and ovarian cancer. BRCA is implemented in a web tool called Canris. I'll read that, www.canris.org. And it incorporates everything, and it'll calculate an individual's breast and ovarian cancer risk based on everything you know about and all the risk factors and genetics that we know. And that tool is very widely used in around the world, apparently cancer clinics, and they're currently developing a sort of head-down version that will be more suitable for use in general oncology and even in primary care, using this information. GPs have less time to get all the information. But where we've got to with breast and ovarian cancer, so ovarian cancer is clinically implemented. Just to give you a sort of feel for how the comprehensive cancer risk models might go. So this graph is showing on the x-axis an individual's lifetime risk of ovarian cancer, and their probability density curves. And so for ovarian cancer, the purple line on the left is the lifetime risk as population on average, a little under 1%. And the sort of dark red line is the average risk for a 20-year-old pathogenic variant carrying BRCA2 who has got one affected first relative here, the mother affected with ovarian cancer. So her average estimated risk is just over 20%. But if you now superimpose on top of that kind of polygenic, poly-lifestyle risk factors, we get the curves underneath. So the yellow curve is the distribution of risk for that woman depending on the polygenic component. So you'll see that her lifetime risk may be as low as 10% or maybe over 30%, depending on her polygenic risk. And then the green line is based on risk factors, lifestyle risk factors. So it's not a nice smooth curve because they are discrete risk factors, and so it's not important to kind of give you a binomial distribution. But you now get risks less than 10% and over 35%. And if you put the polygenic risk and the lifestyle risk factor together, you get the blue curve. And now you get a substantial number of BRCA2 carriers who have a lifetime risk of less than 10%, and some of the lifestyle risk of less than 5%. And so you can see how the comprehensive risk model may be clinically relevant to these women. So if you're a 30-year-old BRCA2 carrier, you're thinking about your reproductive wishes, sort of against the benefits of having prophylactic surgery. If you're up at that end, you may be more inclined to say, well, I'm going to go for prophylactic surgery and whatever my family size is now, that's enough. On the other hand, if you're down at this end of the curve, you may say, well, actually, my risk is quite low. Wait 10 years or 15 years or 20 years and prophylactic surgery at a later date. So it's starting to make a difference in terms of decision making for the BRCA2 carriers. And you can do the same with RAD51C, do the same with breast cancer risks, BRCA1, BRCA2, and so on. So that's the kind of clinical relevance. And that is pretty much the end of the story for where we've got to with ovarian cancer genetics. What the future is going to be, I'm quite so sure. But we've been finding more and more genetics over 20 years. And one thing I didn't mention is actually some of these variants have implications for therapy. The only new therapy for ovarian cancer that's come along in the last 40 years came directly from GMO genetics, that's PARP inhibitors, which you may have heard about. But we now know that comprehensive multi-risk factor polygenic models actually generate differences in risk across the population, but in some subgroups of the population may be clinically important and clinically interesting. And I will take any questions. Thank you.
Thank you for a very comprehensive and overwhelming story there. So we do have some time for questions. I think our refreshments have actually been done to hallway, so we don't need to go down the hall. So we can take questions from the chat, from external people, or questions from the room.
So I have that is a question. So many discuss what she was, but in the cancer, the breast cancer she was. So in a typical breast cancer she was, BRCA1 and BRCA2 included as co-variant about she was, or still a single variant she was.
So I mean, breast or ovarian, so we don't include them. We don't have BRCA1, BRCA2 status for most. I like the ovarian cancer, ovarian cancer consortium. He was 26,000 cases, 40,000 controls, 100,000 controls. Of those 26,000 cases, we've got BRCA1 and BRCA2 status on found because we haven't got around to do part of the sequencing or any other sequencing on bigger numbers. So for breast cancer, the numbers would be more. We've got BRCA status on something like 20,000 cases in breast cancer, but it's still, a small fraction of the total number. I'm not sure that it would make any difference because most every variant we've found to be associated with ovarian cancer or breast cancer population is also associated with risks and calories. The multiplicative model works really well. It's extraordinary. There's a couple of variants that we found that affect risk and carriers that don't affect risk and population. Not the other way around.
Yes, that they follow up question. So they comment on the interaction between BRCA and BRCA2 is there. Rest of the individual variants are PIs.
It appears to be multiplicative. When you do it, multiplicative model just seems to work well. And I think we were perhaps a bit surprised about that. I mean, people used to think that, well, BRCA1, you kind of have a threshold effect and you can't get a higher risk than that. But in fact, it appears to be not true. You actually get a multiplicative effect. Of course, if you start off with a relative risk of 50 and you multiply that by 5, and then get a relative risk of 250, it doesn't make that much difference to the penetrance because it changes the shape slightly. People get the disease early, but it's for cumulative risk. You can't get a cumulative risk of more than 100. Obviously, your estimates as people get older and get a bit less precise because you don't have so much data to start. But the multiplicative model does work extraordinarily well.
I have a very simple question. So can you go back to the slide where you have confidence interval around the point estimate on slide?
The RAD51 or for the This one, yes.
I'm wondering why, like, it seems that the weights of this confidence interval seems to monotonically increase with the age increases.
So that's just a model. We don't have very good age to see. So to plot that, I mean, first of all, take the point estimate. You know, to plot that curve, you need good age-specific estimates of relative risk. Because all I've done here is apply a relative risk to the population. So the population curve is nice and smooth because you consider the ovarian cancer increases with age. You get the penetrance estimate forever, you have to apply a relative risk to that. You need good age-specific estimates to really know what the underlying shape is. We don't have age-specific estimates because there's just too few carriers to get that. So that's a kind of a model. It might not be right. And so that's assuming that the same relative risk applies to all ages in almost certainly wrong. But we haven't got better data. Is that what you mean?
Yeah, it's a related question. So it seems that so using a lot of case control study designs, and for example, population estimate, have you thought about using family design and that be more accurate for this problem? Because I guess here you just make relative risk estimate and you plug in the incidence in the population. But you will get maybe something more accurate if you stability.
Yes, so for example, there is kind of international efforts being made by PALB2, put together all the case control data with all the family data where people have done genetic testing within families and good pedigree data. It does add a lot, but it's hard to get those data on numbers. So, for example, for PALB2 breast cancer, we have much better data and better estimates of the age-specific risks of both breast and ovarian cancer. For RAD51C and D in ovarian cancer, that sort of effort is currently underway. People trying to gather all the RAD51C and D carriers. I mean, one of the issues has been that these things haven't been tested clinically until very recently. So people have only just started identifying families where these variants are segregating. The answer is absolutely yes. We know that when we put all these data together, we'll get better estimates and that in our kind of process. It's a slightly disingenuous slide because I know that we've got better estimates now, and I could some of them quite confidence on there that we know already, but it's kind of more there to make a point about needing from integrated epidemiology in terms of from association with reports.
Thanks, Rob. Thank you very much. So I had a couple of questions that I start with the first one. First one regarding the prevalence of a particular genetic mutation or it looks the time, right? So it obviously related to the age at which we are both the reproductive age and downwards very fast. But do you have any, and I would assume, I would look at it, I would say prevalence, a product of prevalence and risk would determine the speed of development of the next option. So since I'm not in the field, it might be something very obvious to all of you.
Well, firstly, for that, I mean, these disease risks, most of the risk is after reproductive age. So there's always a theory. So to some extent, kind of selection is not a big factor. And these variants are rare. They've not been around the selection is enough so that they haven't been, there aren't common BRCA1 carriers. And so there are reasons that haven't become common. So lots of, when people have been able to estimate the age of mutations, I get this number wrong, the common variants in the Ashkenazi population have been estimated to be about 5 generations old, quite old. Some of them You know, the allele frequency of individual variants is extremely rare. And total allele frequency of truncated mutations of BRCA1 is about 1 in 1,000. There's hundreds. So the individual frequency is 1 in 10,000, 1 in 100,000. So the individual alleles are very, very rare. They've occurred relatively recently by de novo mutation.
It's not yet computed. I understand that. But another question related to this point of multiple genes being involved. I know you said that these are not edited or multiplicated. I'm not sure what the but I don't understand why it is not in fact. It's not. I mean, it is edited and multiplicated. So I'm just there should be situations where in a protein pathway, the one protein being affected should influence how the other protein behaves. So is it that these targets all work in complete isolation that would be necessary?
So I think a really good example of that. So in double-strand, there's all of this by saying I'm not a biologist. There's kind of second-hand knowledge, but you know, BRCA1 and BRCA2, RAD51C and RAD51D are involved in double-strand break DNA repair. And it's involved in the homologous recombination of double-strand break DNA repair. And you know, from what we know, these proteins form one big complex, lots of proteins all gathering around the broken DNA, fix it. So some parts of them are involved in recognizing the break, other bits of it are involved in repairing the break. So in some sense, you know, the pathway always, to me, implies you do this, then you do that, then you do something else. In fact, it's just one big multi-protein protein. And why, you know, affecting this one, why there isn't a limit to the effect? Well, if you've got BRCA1 variant, why does having a RAD51 variant make any difference? Because but it seems that it does make a difference. We don't have very much evidence that like those rare variants interact. Because they're rare. People who carry a RAD51C mutation and BRCA1 mutation are just like super rare, so we can't really say for sure. But there is some evidence for people who carry a BRCA1 and a BRCA2 mutation. They are slightly higher risk.
I feel like the role right here.
I hope it's all the role.
Maybe you can comment a little bit on the extent of allelic heterogeneity in some of the pathogens and differential penetrance across variation dimensions.
So there's quite a lot of data on allelic heterogeneity of BRCA1 and 2. I mean, that goes back to very early days of the work of BRCA1 and 2, showing that the location of a variant dependent on the protein. They're all protein truncating variants, but depending on the position, the relative the risks of ovarian cancer and breast cancer were relatively different. With time in ovarian cancer cluster regions, so variants in the middle of the BRCA1, for example, see how a relatively bigger effect on ovarian cancer compared to the breast cancer risks. So there's definitely a allelic heterogeneity. Then if you go down to non-truncating mutations, there are, for example, there's a polymorphism in the internal exon of BRCA2 that is associated with small increases in risk, like relative risk of 1.3, nothing like the relative risk of 20, 30, 40. The other truncating mutations occur. So there's definitely allelic heterogeneity. There's nowhere near enough data to say that for RAD51C because we've only got a handful of carriers. I think there's some of the families, once you get large family series from family cancer clinics, and you'll be able to start to say whether that's making a difference in terms of to specific cancer risks, but we don't have that information. I mean, some of the other genes, I mentioned ATM because it's breast cancer, not sickle cell and ovarian cancer. So variants in ATM are associated with breast cancer, definitely big differences in risk depending on variant in ATM. But you might expect that when it's a missense variant, because missense variants, some of them have no effect at all, and some of them seem to be like truncated mutations. Truncated mutations, it's much harder to show.
I'm gonna leave it now the big role here. I do have a question that I'll pass on to you. You're one of the first person that ordered the concept of polygenic restore on your the New England Journal paper. Decades before the PRS branching, right? So PRS is really, it works really well, is what you're saying. The issues right now is implementing the PRS. And one of the issues and front and center is ancestry differences. So the movement is scientifically, it is important to do ancestry specific PRS because we know they are different. But when you're implementing, it's incredibly difficult to implement ancestry specific PRS in the diverse population such as in Canada. So what is your take on how do we tackle these issues for PRS for implementation?
Well, firstly, there is, across lots of different disease phenotypes going way beyond ovarian cancer. So I've argued that there's some clinical implementation that's relevant in ovarian cancer, and same in breast cancer, mainly in carriers of rare variants in the other higher risk genes like BRCA1, 2, RAD51C, PALB2. I don't think there's much place for PRS as a general population thing. My view is at the moment, the only kind of population PRS that might be useful is coronary heart disease. I think there is good clinical reasons to use polygenic scores of coronary heart disease because of the way we manage coronary heart disease risk with medications and so on. So, addressing that, there's not much clinical implementation. Ancestry specific. The first thing to say is that a PRS that's based purely on individuals of white British origin, just to really narrow it down. That will perform reasonably well, depending on how good it is in that population, but it will perform reasonably well in lots of other populations. It's not like it doesn't work at all. It's the first thing to say. So, obviously you want the best possible score in any given population. But there's lots of work going on now where you can use all possible data to get multi-ancestry scores. And you can, you're genotyping somebody, you can do your principal components and put them anywhere on your principal component map you like, and then know how your polygenic score applies to those individuals and give them a reasonably accurate risk estimate. The other thing I would remind people, when you give somebody a risk estimate, say your risk is 20%, for sake of argument, you get that wrong 100% of the time, because that person either gets the disease 100% or they don't, 0%. Nobody ever gets 20% of a disease. And so a risk estimate is a very kind of odd thing. There is no, you don't say you will get the disease and you're right, or you will get the disease and you're wrong. You say, well, you've got a 20%, so you're always right or you're always wrong, depending on how you look at it. So it's only how well it performs in a population, and it performs with certain characteristics. So I don't think it's a big issue. Obviously, you would like better risk predictors in different populations, but we've got risk predictors, they work better or worse. We want to keep making them better.
So I have a more technical question. And so you talked a little bit about the clinical ability or platform just now, but for a tool like the more comprehensive tool that you showed us the website of that includes the PRS and maybe some of the other less penetrant genes. And I guess this is a question about risk prediction more generally. What sort of evidence currently exists or that you think should be generated to show that having patient care informed by this risk prediction will result in a benefit for the patients in terms of things like survival outcomes and quality of life and things like that.
Well, that's a whole lecture in itself to answer that question. So, you know, people always like to say, oh, you need randomized controlled trial evidence. And, you know, for example, you know, we don't have randomized controlled trial evidence that testing cholesterol is a good thing. Nobody's ever randomized people to you get a cholesterol test, you don't get a cholesterol test. Now we're going to compare long-term mortality. The only randomized controlled trials we have is, well, we do a cholesterol test and then they've been randomized to treatment. And so the ultimate randomized controlled trials on prevention are almost never done because we kind of assume that we can sometimes extrapolate from. And so, you know, nobody's done a randomized controlled trial of bilateral salpingo oophorectomy in ovarian cancer. It's risk reducing for ovarian cancer. It's the whole thing like that, the randomized controlled trial of parachutes or BSO is like a parachute of somebody's high risk of ovarian cancer. So it becomes a very difficult thing as to when you need RCT evidence and when you just assume. I'll use the example of chronic heart disease. So, you know, we know pretty much that you can give anybody a statin without knowing anything about them at all, as long as they're human. And it may even apply across species for all I know. But we know that giving almost anybody on the planet a statin will reduce their risk of dying of chronic heart disease. The randomized trials were huge. It didn't matter what their initial cholesterol was, it didn't matter whether they smoked or not, it didn't matter what blood pressure was. Secondary prevention, primary prevention, statins reduced everybody's cholesterol, and there's no threshold which having your cholesterol low doesn't reduce your risk of chronic heart disease. So the only reason why we don't have statins in drinking water is because of the kind of rare side effects of statins and, you know, the cost. And so we don't have any trials of risk stratification studies. I can only say, you know, what people do in the US, but in the UK, GPs are supposed to offer statins to individuals above a certain absolute risk of chronic heart disease. Use different algorithms for estimating that risk. Q risk is a common one. But nobody's ever done a trial where they say if you use Q risk or not, does it improve your outcomes? Because we assume that the randomized trials of cholesterol lowering will apply some general sense, we can model the benefit. So I think the same is true of PRS, polygenic scores, that, you know, it's the intervention that matters, not PGS. So the idea that you need to randomize people doing PGS or not is just a total nonsense. There are some things you can do without trials. And if you can stratify people on risk for cholesterol based on blood pressure, then why can't you do it based on polygenic scores? Why treat polygenic scores?
One more question.
I have a question about the independence of the conventional risk factors in PRS. So you kind of assume that they're independent, but we know that there's a lot of pleiotropy from many different genetic studies. Is there any evidence that the risk factors are associated with some of the variants in PRS?
Each one about ovarian cancer specifically, or every phenotype or so we've looked within the Simba consortium, the consortium of BRCA1 and BRCA2 carriers, has looked at the modifying effect of the well-known risk factors for breast and ovarian cancer. And yeah, all we can say is that more or less it appears to be multiplicative. I mean, you know, your estimate of the relative risk in a carrier versus a non-carrier, they're never exactly the same, are they? And, you know, demonstrating for sure that this relative risk is 2.2 and this one is 1.9 is very, very difficult. But more or less, it seems true. And what you can do is show that if you look at a sort of polygenic, you know, you can't rule out this SNP interacts with this risk factor. But it's much easier to say that this polygenic score interacts with this multi-lifestyle score, and it really does appear to be close to a multiplicative model. When people have looked at the kind of cumulative, you estimate your polygenic epithelial factor score, and then you redo your case control analysis, stratified by polygenic score. The estimated odds ratio in those groups is very close to the predicted multiplicative model. I would say apart from right at the tails of the distribution, where you seem to break down a bit, but you might expect that. So across the vast range, it seems to work. But that would be very hard to say, but for an individual, well, you just have to have, 2 copies of this allele and you have endometriosis, but that doesn't suddenly mean you're a very high risk of ovarian cancer. That kind of those unusual things you can't do. But I'll go back to the fact that your prediction model wrong for everybody, it just might be a little bit more wrong for those people than others.
To close off the questions, so people can continue to refer to my questions, and we have some more questions, and so people are talking to stay. So let's thank our speaker again.